everybody to be here. And I would like to thank the organizer for inviting me to talk about some of the research that we perform at the Institute of Ciencias Nucleares at UNAM. As the title is, the possible role of clay minerals in chemical evolution process. The outline of the talk will be a brief introduction, ionizing radiation and mineral surfaces, absorption, irradiation of prototype compounds. Life is a particular, a unique system, as a continuation of the evolutionary process of the universe. It is a complex machinery, at the same time as familiar as it is mysterious, being an ensemble of millions of molecules. How life was originated is one of the most difficult questions in science. Other questions associated with this are, was the unique event? Are we alone in the universe? So, the fascination of humanity in the origin of life has a deep root. This is a multidimensional subject that began as a religious and philosophical issue, but that's later taken up by science. What are the minimum properties required for a cell? A consensus view of the necessary characteristics that a system should possess are three: metabolism that gives energetic autonomy, compartments that gives individuality, genetic machinery that gives history. All these systems need organic compounds to function. So in order to have all this, we need abiotic synthesis of organic compounds. So how living beings were originated, how their molecules were formed and organized in structures? One way to get an insight into this question is by simulating the processes that may lead to the origin of life. So from the chemical point of view, we need to build chemical models that were based in astronomical, geochemical, and biological observations. So the simplest working hypothesis for these studies are that some molecules of biological importance for life at the present also were important at the origin of life. So some considerations that are necessary while doing simulation experiments is to consider the raw material that we will use, the energy sources, plausibly geological environments, and the fate of these molecules after a while. So, which molecules? How abundant are they? Do they resist environmental conditions? These are questions that we need to take in account to build a simulation experiment. Energy is responsible to start, to promote, and to direct all these physicochemical processes. So it's a fundamental tool in these studies. There are several sources that have been proposed to carry out these processes, but they need to have some characteristics. They should be abundant, accessible, and efficiency in the primordial times. For solid-state reaction, is also needed highly penetrating energy sources, available in geological scenario, efficient for inducing chemical reaction. Among the possible sources of energy, ionizing radiation meets all these requirements, and I will focus on this. There are two sources of ionizing radiation on the Earth. One are external, it came from the cosmic rays, and the internal ones that are because of the radionuclides that were formed since the Earth was formed, and we can divide in three groups. The existing up to now that are radionuclides with half-lives in the order of 10 to the 7 to 10 to the 9 years. Another group are probable sources that they are the natural nuclear reactors and the radioactive products like the Okla phenomena. And the third group are the possible sources that are the super heavy elements with atomic number of 112 to 124. Ionizing radiation and also other sources have a dual performance. They can be an angel because they help to desynthesize of organic compounds, but also as a devil because they can destroy if the interaction is for a long time. So we need to take account also geological environments, because a simulation of the early Earth's possible environments may require the participation of a multi-phase system. So we need to have raw material, a surface, and a tool that is generous. The possible roles of minerals in chemical evolution can be summarized in three: desorption, that helps the concentration of the molecules; the catalysis, to take care of some reactions; and the protection to external source of energy. But which are the minerals that can be considered? The most geologically relevant mineral surface present on the primitive air were carbonates, silicates, sulfates, and clays. So I will focus on clays. They are hydrated aluminosilicates weathering from feldspats. Clays are built from silicon tetrahedral layers and aluminum octahedral layers. According how these are arranged, they give rise to different clays. In particular, I will mention this, that is sodium mormonite, that is of the family of smectites, and is built with a tetrahedron, octahedron, a tetrahedron layer. This is the cell unit. Between cell unit and cell unit, there is a space called interlamellar channel. This surface present a negative charge both, so we need some cations to be here. The IHs of these are negative or positive according to the pH. So we will focus on the stability of some prototype compounds absorbed in minerals and exposed to a high radiation field, to study the possible protective role of minerals like clays, and also to highlight the importance of ionizing radiation in chemical evolution processes. We need to consider some things while doing simulation experiments with clays. First, if there is an absorption, the extent of the absorption, the site of binding, and the mechanism of absorption. So why is important the site of binding? Because the catalytic behavior depends on the Lewis or Bronsted sites in the clay, and also the protection toward irradiation can be related to the site of absorption. So essential bioorganic molecules in chemical evolutions are needed, but some of them can be very unstable in the conditions of the prebiotic earth, so mechanisms for stabilizing them are required. As important is the synthesis of prebiotic compounds as their stability under those environments, so should be a balance between the synthesis and the degradation of the compounds. The stability of organic compounds in the prebiotic environment can be from different sources. One can be that the molecules is very stable in those conditions. Second, perhaps is a continuous synthesis, so always we have this molecule. And the third is a protection mechanism from several external sources. For example, by the interaction with a mineral surface. So again, the possible roles of clay minerals in chemical evolution as proposed by Bernal are these three parts. The first is desorption, that helps with the dilution problems. The catalysis, that, for example, in the laboratory, we make some catalytic functions with the clays with carboxylic acid, and it produces biased roots. And the third one, that is the protection route for high energy radiation from an external source. I will focus in this part of the protection role. So what are the questions we wish to address? Do the clays are capable of absorbing organics? To what extent? Where? Do clays offer protection to the molecules that are absorbed onto it? If yes, do you mean for all the compounds? Do clays catalyze reaction in a preferential way? And all the reactions with different energies are the same? For this last point, I will exemplify with this. This is formaldehyde molecule and it's absorbed at 250 nanometers. This is in the outer part of the atmosphere. When it falls down with the rain and gives to the aqueous body, immediately is transformed in the hydrated form. The hydrated form is not absorbing at this wavelength, so it's a way of preservation. But if this hydrated form is near to an ionizing radiation source, well, it will disappear also. So the type of radiation is important, and it's important also the balance. To study the possible protection role of the clays, we study prototype compounds to simplify and make a systematic study. So we choose purines and pyrimidines because their synthesis and stability in environmental conditions are of paramount importance in chemical evolution. They are formed in periodic experiments. So some of the results that we got are the following. First, we need to study the absorption as function of the time, the stain absorption, and the binding sites. So the experimental part was dividing first the absorption, the radiation, and the analysis with mainly with chromatographic techniques. The absorption experiments depends on the nature and structure of the sorbent, the structure of the absorbate, and the pH, the type of interaction that is carried out, and the presence of a co-sorbate. So I will present some of the results with these experiments. This is the effect of pH in one of the bases that we study, in one in specific. As this figure says, the maximum absorption is when we have a acidic pH. And as the pH increase, the absorption decrease. So this region is the best for our studies. This table summarizes some of the results with purines and pyrimidines. As you can see, adenine, xanthine, and guanine, in general, purines are almost 100% absorbed on the clay. But it's not the case with pyrimidines, with the exception of cytosine that is absorbed 100%. Thymine and uracil are little absorbed or not absorbed at all. These results can be explained because the species that are present according with the pH. So we make this speciation, we see that according with the pH, we have different regions in which different species are dominating. For example, for adenine, we have at pH acid, we have this cation weight. So the same happens with guanine and cytosine, but the thymine and uracil at that pH is neutral. So these cations are entering in the interlamellar change, but in ionic interchange, and occupy this space, and they are absorbed. So there is a coabsorption phenomena that is very important in these studies. When a molecule is really absorbed in a solid, for example, adenine, may have synergetic effects in a second molecule, for example, uracil, that enter in the clay. And this second molecule now is absorbed. For example, we make a solution with adenine and uracil, and these are the results. As you can see, the absorption with the time, it has a very fast absorption after it's already equilibrated. If you see, adenine is absorbed at the same percentage that was when it was alone, but uracil was not absorbed and now is around 45 absorption. So this means that with the coabsorption, some molecules that by themselves they are not absorbed can be stay in the clay. The site of binding is also important, and we took two strategies to see in which place of the clay the molecules were bound. So these three X-rays. This is the untreated clay in which this distance corresponds to this angle that is the cell structure. When we treat the clay, the morning or night, with this molecule that is an quaternary ammonium salt with a long change, all these compounds bound preferentially in the active sites of the clay, forming these structures. So the distance between unit cell and unit cells increase, and we can see by the angle. When we treat the clay with a polyphosphate, the polyphosphate has negative charges. So at this pH, the edges are positive. So here, the bounding of polyphosphates. Since we don't interfere with the distance in between the cells, the interlamellar channel angle is still the same. So these are the results with blocking these channels. This figure represents the untreated clay that is almost 100%. But if we put this one in with the treated clay blocking in the interlamellar channel, we have these results. There is a notable decrease in the absorption. If we make the experiments with the phosphate clay blocking the crystallage, there is no modification in the absorption. So with this, we can conclude that guanine is sorbed in the interlamellar channel. In the case of poly-A, we repeat the experiments and we find only when we block the edges, the absorption decreases considerably. So in this case, poly-A is bounding at the edges of the crystal. Well, we performed some experimental with radiation to see if the clays were able to protect the compounds. So the question was, organic compounds that have been absorbed into the clay minerals and irradiated with a high radiation dose, can they be recovered from the clays? In other words, the clays can protect the organics absorbed on it. So these are the results of this experiment to solving these questions. Here, there is the response of aqueous solution adenine without only aqueous solution. As you can see, it is almost destroyed in less than 30 kilograms of irradiation dose. But if we absorb this solution in clay, the recuperation of the adenine increase very much and it can resist very high radiation doses. But if we take this system with the clay, the adenine, and we dry it, and we observe that the increase of the stability to high radiation doses. And if we took out from the clay the adenine, this is still adenine. So it means that the clay really can protect from external doses the molecule. With the pyrimidines was also similar with cytosine. This is a very light molecule that decomposes very fast in aqueous solution, but with the clay, the survival increase. Thymine is not absorbed at all in the clay and is very labile. And the amount that is sorbed is at the edges, so it was not protected. So these are the results for the coabsorption radiolysis experiment in which we observe the clays can protect the molecules that are sorbed on them. So what is the mechanism is currently under study. A possibility is an energy transfer from the clay to the absorbed molecule. So it is important to remember that we need a balance between accumulation and degradation. If the degradation is high, all the products will hold, so we need to take it into account, and minerals can be the difference. As final remarks, clays are well distributed, have an ancient origin, and they are abundant. They have been detected in extraterrestrial bodies too. Sorption is a fast process. The sorption of the prototype compounds is better at acidic pH. Not all the compounds are sorbed at the same extent and depend on many factors. And their role as protecting agents under plausible periodic conditions can be possible, but not for all the molecules. Other experiments that are carried out in our laboratory show the catalytic properties in processes related to chemical evolution, in particular to bias synthesis. So I want to thank my colleagues in the Institute of Ciencias Nucleares and the students related with this laboratory, which are the essential parts, and the data that I present, it was part of them. And I want to thank you, all of you, for your attention.